Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the topics within the biology for the class 11 and 12 under the program IIT PAL. And in this series, we were discussing about the biotechnology principle and processes. And if you recall, in our previous couple of lectures, we have discussed about the biotechnology and its potential to develop different types of products as well as to solve the existing problems. And what we have discussed that the biotechnology is using two approaches to solve the problem or to develop the new product is that it is either using the genetic engineering protocols to design the genetically modified organism, or it is utilizing the bioprocess or the bioengineering approaches to amplify or to enhance the product productions. So in this series, we were discussing about the genetic engineering, and the genetic engineering is involving the different types of procedures where we are using the different types of tools. So in our previous lectures, we were discuss about the different tools like polymerase, diffusion enzymes, ligases, and vectors. And in our previous lectures, we also discuss about the isolation of genomic DNA, as well as once you have isolated the genomic DNA, how you can be able to analyze that onto agarose jelly electrophoresis. Now in today's lecture, we are going to start our discussion about the new topic, and that is the polymerase chain reactions. So the polymerase chain reaction, as the name suggests, it is actually polymerization reactions which are actually going to be run to synthesize the new DNA or the new chain of nucleotides, which is always being done with the help of the polymerase enzyme. So that's why the name of this the reaction is called as the polymerase chain reactions. So what is the polymerase chain reaction is doing? It is amplifying a double standard DNA with the same size and sequence by the enzymatic method and in a cyclic conditions. So if you recall, we have discussed about how the polymerase chain reaction can be used to isolate a DNA fragment from from the genome of an organism. So you can imagine that if you are isolating a genome from an organism, it could have the different types of genes like gene 1, gene 2, gene 3, gene 4, gene 5, and gene 6, and so on. And suppose we want to isolate the gene 1, what we can do is we can use the DNA polymerase, and we can put it into the polymerase chain reactions, and that is actually going to allow you to amplify the gene 1. But the question comes how the polymerase chain reaction actually allows you to amplify the gene 1 and how you can be able to perform the polymerase chain reactions. But before getting into the details as well as the complicated process of polymerase chain reactions, it is important to recall what you know about the simple molecule like DNA. So DNA is a nucleic acid, and it is composed of two complementary nucleotide binding block chains, which means you are going to have the double stranded molecule where you can have the two strands, one is on this side, the other is on this side, and where you can have the nucleotides on the each strands, and each nucleotide is facing the another nucleotide on the other strands, and that's why they are actually making a complementary base pairing. The nucleotides are made up of the phosphate groups, a carbon sugar, and a nitrogenous base. The DNA has 4 different types of nitrogenous base. It has the 2 different types of purines like the adenine as well as the guanine, and it has the 2 different types of pyrimidines, the cytosine as well as the thymine. These 4 bases are linked in a repeated fashion by the hydrogen bonding between the nitrogenous bases. The linking of the 2 complementary strands is called as the hybridizations. And if you can recall, there is a definite pairing between the purine versus the pyrimidine. So the A, which is actually a purine nucleotide, is always making a base pair with T, and the number of hydrogen bond what they share is the number of hydrogen bond what they share is 2, and compared to that the G, which is a purine, is making a pair with C, and they are sharing the 3 hydrogen bonding, and this pairing of A to T and G to C is very consistent, and that's why the two strands of the DNA is complementary to each other. Now, what is mean by the complementary to each other? For example, you can understand that by this example, that where you have a primary sequence. So for example, you have a sequence of GGC, TAT, GT, GA, TTC, and this is a strand 1. So if this is your strand 1, and you know that A is going to make the pair with T with the help of the 2 base pair, and the G is making a pair with C with the help of the 3 hydrogen bonding, then what you can do is, if you have to generate the complementary strand, wherever you have the G, you can actually put the C, and wherever you have the A, you can put the T. So that's why if you have, this is the primary strand, you can easily generate the complementary strand by C, C, G, A, T, A. So what you see here is, wherever you have the G, you are actually having the C onto the complementary strand. So that is what is called as the complementarity within the DNA sequence, and that actually is a very helpful feature, because it allows the system to know what sequence he has to synthesize. For example, if we are getting this sequence, we know that this is the strand. If we synthesize, then you are actually going to get the complete DNA strands. So this information is good, but what about the machinery? So DNA polymerase is the machinery which is actually going to utilize this information, and that's how it is actually going to synthesize the complementary strand. So if you have a one strand, the DNA polymerase is going to sit onto this strand, and that's how it is actually going to synthesize the complementary strand, utilizing the information what is given onto the top strand. So because both the strands are complementary in each other. But you know, and if you recall, when we were discussing about the polymerases, the DNA polymerase does not work in isolation, because that there are multiple steps which has to be performed to perform the DNA synthesis. What are these steps? You have initiation steps, where you are actually going to prepare the DNA. And if you recall what we have discussed, the DNA, first of all, the DNA is a double helical structure. So this double helical structure has to be this helicity has to be removed, and that is being done by the helicase. And then the two strands are going to be removed from each other, and that's how the nucleotides are going to be exposed for an enzyme to read. And once the DNA is being present as a single standard DNA, then this single standard DNA has to be maintained as single standard DNA by another enzyme which is called as another protein which is called as the single standard DNA binding protein. And these two events, where you are converting the double helical structure into a single standard DNA and then maintaining the single standard DNA so that the synthesis will go into the next phase, is actually are called as the preparative phase, and that will be the initiation stage. Once the DNA is present as a single standard DNA, then the DNA polymerase will come, it will sit onto this strand, and then it will read each and every nucleotides, and that's how it is actually going to start adding the complementary nucleotides. And once it reaches to the end of the strand, it is actually going to be dislodged from this particular complex, and at the end you are actually going to get the two strands, or you are going to get the because you are starting with the two templates, so it is actually going to give you the two molecules. So this is all about the DNA synthesis if you utilize the DNA polymerase. But DNA, as I said, DNA polymerase cannot synthesize the DNA in isolation. It requires the additional protein under the natural conditions. But providing these proteins as well as the properties is very difficult, and that's why the scientists have started discovering the conditions under which you can be able to replace the function of helicase as well as the single standard DNA binding protein in such a way so that you could be able to utilize the DNA polymerase directly with the help of the single stranded DNA. And that is what is being done when the people have discovered the polymerase chain reactions. So what is happening in the polymerase chain reaction is that, for example, you have a double stranded DNA. So you know that if you have a double stranded DNA and if you are actually going to heat this DNA to certain temperature, the heating will actually going to destroy the hydrogen bonding between the A and T, as well as it is actually going to destroy the hydrogen bonding between the G and C. So once the hydrogen bonding is going to be broken from the two strands, these two strands are actually going to be get separated and they will form the single stranded DNA. But once these two will form the single stranded DNA, then you actually can allow the primer binding. So you can add the primer and the primer will bind onto the this strand as well as onto the this strand, and then you ask the enzyme to sit onto this particular template. So one enzyme will sit on this, one enzyme will sit on this, and that's how the enzyme will start adding the nucleotides in this direction. This enzyme will start adding the nucleotide in this direction, and that's how after the one cycle you are, since you started with one strand or one templates, you are actually going to end up with two templates. One is your parent DNA, the other one is the synthesized DNA. So this is what is showing here, that if you started with the double stranded DNA and if you denature the DNA with the help of the very high temperature, the DNA is going to form the two strands. So one is this template, the other one is this template, and after that what you have to do is you have to add the primer and you have to add the enzyme. So once the primer is going to bind, it is actually going to bind onto the complementary sequence. So primers are actually the DNA sequences which are actually having the complementarity onto the corners of this particular strand. So once it will bind to this particular DNA, the enzyme will recognize this primer dimer this primer template complex, and that's how the enzyme will sit and it start synthesizing in this direction. Once it reaches to the end of this DNA, the synthesis will enter into the termination stage, and that's how you are actually going to synthesize the two templates. So you started with one template, for example. So in the cycle 0, you started with one templates. After the first cycle, you are going to have the two strands. Once you continue, so if you continue this whole cycle again and again for many cycles, so for example, for first cycle you are going to started with 1, you are going to have 2. After the second cycle you are going to have the 4. After the third cycle you are going to have the 8. So after every cycle it is actually going to be double. For example, in fourth cycle it is going to be 16 and so on. So that is what is called, that's why it is called as the polymerase chain reaction, because here you are putting the things in a cyclic event, like first the DNA denaturation, then primer diming and elongations, and then termination. And this will continue as many cycles you want, and that's why this whole reaction is called as the polymerase chain reaction, because it is polymerizing the nucleotides in a chain like events, and that's how it is actually synthesizing the new strands of DNA. So the PCR is a repeated cyclic reaction that involves the mechanism of DNA replication. It results in the production of multiple copies of DNA from a single one. The whole process involves three main events: the denaturations, annealing and elongations, and a DNA fragment of your interest is used as a template from which a pair of primer or the short oligonucleotide complementary to the both the double standard of DNA are made up to prime the DNA synthesis, whereas the direction of synthesis or the extension is from 5 prime to 3 prime as in a DNA replication. The number of amplified DNA or the amplicon increases exponentially per cycle. This means one molecule of DNA will give rise to 2, 4, 8, 6 and so forth as many cycles you go. That's why if you want to calculate the amount of amplified DNA, you can use this particular type of equations, and where the C is the final amount of DNA, the C naught is actually the initial amount of DNA, and E is the efficiency, N is the number of cycle, and S is slope of the exponential phase. And that is actually going to give you the complete amplified amount of DNA what you are going to get after the n cycles of the polymerase chain reactions. So what you see here is that the polymerase chain reaction looks very simple, but it requires the multiple steps, and these steps has to be performed in a very precise manner so that you will be able to get the amplified product in a, as accurate as the template strands, and it also should be give you the amplification of the amplification in a high level of amplifications. So what are the things you require? So requirement of the PCR. So first thing is you require the DNA sequence of the target region must be known. For example, you cannot use the PCR if the target sequence is not known, which means the PCR cannot be used for the unknown sequences. For PCR, the genomic sequence as well as the sequence of the gene should be known. Then why that is required? That is required because you want to synthesize the primer. So for example, if I have a gene like this, what I have to do is, if I am to synthesize this gene from the genome, what I need is I need the complete sequence of this particular gene so that I can be able to synthesize the primer. Although primer synthesis and primer designing is beyond the scope of this particular chapter, but just for your understanding, what you can do is primers are nothing but a complementary sequence of this particular gene onto the 5' end. So example, in this case, this is the 5' end, and in this case, this is the 5' end. So if you take the complementary sequences, these two primers, these two oligonucleotide sequences are going to be served for the primer to amplify this particular gene. This particular primer, which is on the top land, is called as the forward primer. So this is called as the forward primer, and this one which is on the reverse strand is called as the reverse primers. These can be readily produced by the commercial companies, can also be prepared using a DNA synthesizer. So once you know that sequence, you can ask the companies to synthesize a primer, or you can actually synthesize using a DNA synthesizer. So that is the primer what you require, and then you require the DNA polymerase. So you know that the DNA polymerase, what Arthur Kornberg has isolated from the bacteria, from the is actually a thermolabile. So what will happen is, when you go with these kind of denaturation cycles, and when you heat the DNA to 95 degrees Celsius, these thermolabile enzymes are going to be denatured, and this means you might have to add the enzyme after every cycle. And that is what people were doing when they discovered the PCR, but they were not having the thermal stable DNA polymerase, which means the tag DNA polymerase. So once the people have discovered the tag DNA polymerase, they started adding that, because if you have the tag DNA polymerase, even if you heat the DNA to 95 degrees Celsius, which is actually going to be the first step of denaturation, the tag DNA polymerase will still remain intact, and that's why you don't have to add the enzyme every time. Then you need a machine where you can be able to perform the PCR reaction, and that is called as the thermal cycler. The machine which can be able to program to carry out the heating and cooling of sample over a number of cycles. This is called as the PCR cycle, and this is the PCR cycle what you have to do. So to amplify the DNA, where you have the multiple steps, like for example, you are going to have the initial denaturation. So initial denaturation you have to do at 95 degrees Celsius, and that is actually going to convert the double standard DNA into a single standard DNA. Once you have done that, then you are actually going to allow the enzyme to go for the denaturation, annealing, as well as the elongation. And these three events are considered to be one cycle. So in the denaturation, again, you will denature the DNA before you start annealing step. So this is first step in which the double standard DNA will template is denatured to form the two single standard DNA by heating at 95 degrees Celsius for 15 to 30 seconds. Then what you do is you cool down the reactions. So you cool down the reactions so that the primer what you have added should be able to bind the disc templates. So that event is called as the annealing step, because the binding of the primer to the DNA is called as the annealing step. So this is the annealing step, where at lower temperature, usually the 50 to 65 degrees Celsius, the primers are allowed to bind the template DNA. Annealing time is 15 to 30 seconds, and it depends on to the length and the basis of the primers. So this, depending on the length of your oligonucleotide primers, you can actually set the annealing temperature. Now, once the annealing is over, then you can increase the temperature further so that the DNA polymerase will sit, but the other strand, because apart from this, you are also going to have another strands, and that strand also can actually again form the double helix. So you have to again bring the temperature high so that these two strands should not be able to form the double helix, and in the meantime the enzyme will sit and start synthesizing the new strands of DNA utilizing the primers. So elongation, this is the synthesis step where the polymerase perform the synthesis of new strand in the 5' to 3' directions using the primer and the oligonucleotides what you are going to add. An average DNA polymerase adds approximately 1000 base pair per minute, which means if you have a gene of 1000 base pair, you have to give the annealing temperature of you have to give the elongation time of 1 minute. So accordingly, you can actually be able to decide how long you want to give the elongation so that the enzyme will sit here and should be able to walk up to the end of this particular gene. Uh, so step A, B and C actually makes the one cycle, and this actually will continue for multiple cycles. For example, in this case, in this example, we are saying that this will continue for the 30 cycles. So if this will continue for 30 cycles, you can imagine that if you started with one, it is actually going to give you the lot of amplification of the same templates. After this thing is over, then you are actually going to do the final elongations at 72 degrees Celsius for 10 minutes, and that is actually going to give the enzyme to complete the elongation of all the other DNA strands. And after that, you will ask the machine to keep the reaction at 4 degrees until you will not take out, and that is what is called as the final hold. So after the cycles are complete, the reaction is held at 70 to 74 degrees Celsius for several minutes to allow the final extension of the remaining DNA fully extended. So, and then you keep the reactions at 4 degrees for final holds. This is the way you have to do the polymerase chain reaction setting. For example, you have to first do the initial denaturation at 95 degrees Celsius for 5 minutes, and then you have to do a 30 cycle of uh, the PCR cycles. So initially, so you have to have the denaturations, annealing, and extension, and then you are going to have the final extension for at 72 degrees Celsius for 5 minutes. So this will continue for 30 cycles. So the enzyme, so the machine will first go to this, then it will go to this, and then it will go to this, and then again from here it will go to this, and that's how it will continue this cycle for 30 cycles. And once the 30 cycle is over, then it will go to the the final extension, and once the final extension is over, then it will go to the hold. These are the reactions mixture. So where you going to have the template DNA, you are going to have the primer. So as I said, you're going to have the two different types of primer, the forward primer as well as the reverse primer, and then you have to have the magnesium chloride, the deoxyribonucleotide or the DNTPs, which are actually the building blocks for the DNA, and then you also require the thermostable tag DNA polymerase. Once you have all these reactions set up, and then you can put it into the machine with which is called as the thermal cycler. So this is the machine. What you have is a thermal cycler where you have a heating block, and then you have the place where you can actually be able to place the reactions. So for the PCR reaction setup, you have to have all these components in ice, and that's how you can be able to set up the reactions. So to just give you a very real and virtual experience how to do the PCR and how you can be able to set up the reactions, I would like to take you to my lab where the students are actually going to show you how to set up the reactions, and then how to actually program the machine, and that's how you can, and then they will actually going to do the PCR. And I hope that this particular type of demo is going to be helpful for you to understand the whole process.
Widely used molecular biology technique to amplify a particular segment of DNA. It is also employed in biomedical research and forensic medicine. The main application of this polymerase chain reaction is cloning. To set up a PCR reaction, we need template DNA, site-specific primers, DNTP mix, nucleus-free water, and Taq polymerase. For a 50 microliter reaction, in a typical concentrations of 10 to 100 nanograms of template DNA used and 5 picomoles of each primer will be used. This is an earlier version of thermal cycler which contains display unit where we can observe the parameters and change the parameters. This is a hard shield. This is sample holder, and inside there is a Peltier system which can maintain the temperature fluctuations. For setting up a PCR reaction, initial denaturation at 95 degrees Celsius 3 minutes, and these steps we will use 30 repeats where initial denaturation will be 30 seconds and annealing it extension time should be given 1 minute per kb. And here final extension should be given 10 minutes and hold it 4 degrees Celsius .
Now, once the PCR is over, then you actually can take out the reaction mixtures and then you can be able to do the analysis of PCR product. So once the PCR cycle is completed, the amplified product is loaded into the agarose gel and observed after staining with ETBR under the UV light. A water blank is included to monitor the cross-contaminating DNA source as a template. The percentage of agarose depends on the size of DNA, but generally people use the 0.8 to 1% agarose. So what you have to do when you are trying to analyze the PCR reactions, you are going to run all these reactions along with molecular weight markers. So it is, for example, the DNA markers. So DNA markers are nothing but the DNA of different sizes, for example, 0.5 kb, 1 kb, 1.5 kb, 2 kb, and 3 kb. And then you are going to have the negative reactions, which means you are going to include the water instead of the template DNA, because that is actually going to say that if there will be a contamination of something, and then you are going to have the positive amplifications where you are going to actually have the template DNA. In this way, you see that the template DNA is actually giving you amplified DNA product, whereas in the negative amplifications, you are not getting you're only getting the template DNA which you have added as a and then there's a primer dimers. So because the primers are not being utilized for amplification, that's why you are actually going to see the primers at the bottom of this gel. In this case, for example, the primer are being consumed by the amplification of the DNA in the PCR. That's why you don't see the primers. What is the advantage of PCR reactions is that it is actually very quick, because what you require is you have to require the template DNA primers and the NTPs and water, and then you require the thermocycler, and it could be done. It is very easy. It is just mix and match. Then you put it into the machine, and it is very easy to do. It is very sensitive. Even if you have the DNA at the level of nanogram level, it is actually going to give you the amplified DNA, because you remember that it is actually amplifying in a very because after every cycle it is amplifying the double. So that's why even if you started with nanogram level, it is at the end it is actually going to give you the enough amplified DNA, and then it is very reproducible. It is actually reproducible, and that's why, because of all these properties, the people are using very extensively the PCR, not only for the cloning purpose but also for the diagnostic purpose, because if you have a very small amount of DNA of an infectious organism, even then you can be able to do the amplification with the help of the primers, and you can be able to detect. And that's why the if you might have heard that even for the virus related diseases like COVID-19 also, people are using the primer, people are using the PCR to detect the virus, uh, and the level of virus is very low, and that's why it is very sensitive. So this is all about the PCR reactions, and I hope you might have understood the whole process by looking at the demo what students have shown, and it would be helpful for you to understand how the people are using the PCR to isolate a particular gene from the complete genomic sequence. So once you have done the PCR reactions, you are actually going to get the fragments. Now what you have to do is you have to do a fission digestion and ligations. If you recall, when we were discussing about the rectification enzyme and ligase, we discussed in detail about the rectification enzymes. So now today we will only going to briefly discuss about how to set up the rectification digestions and ligation reactions. So decision digestions, I think we discussed that it should be in a very large volume, like the 50 microliter. So what you have to do is you have to take the DNA, like the amplified DNA, in a 1 microgram. Then you have to put the decision enzymes, if you are doing the single digestion or the double digestion, you can use the one enzyme or both enzymes. Then you have to use the buffer where the enzyme will have the activities, and then you have to use the BSA, and then you also have to use the water, and the final volume should be the 50 microliter. And why it should be 50 microliter? Because the rectification digestion is a hydrolysis reactions where you require the large quantity of hydrogen and the hydroxyl group, and then you incubate the reactions or 12 to 12 to 18 hours at 37 degrees Celsius, and that is actually going to cut your fragment, and it is actually going to give you the sticky ends. Similarly to this ligation reactions, you have to use the ligation reaction with the vector and the DNA. So both are being digested with the scission enzymes, and now vector and the insert has to be taken. The only thing what you have to remember is when you want to do a cloning, the vector and the insert has to be in a ratio of 1:3, which means if you take the vector as 1 microgram, the insert has to be 3 microgram. Then you have to add the T4 DNA ligase and ligase buffer and the BSA, and then finally the sterile water. And the reaction volume should be 20 microliter, and then you incubate all this reaction at 16 degrees Celsius for 16 to 20 hours, so that it is actually going to give you the ligated products. Why we are doing at low temperature? Because so that the hydrogen bonding between the nucleotides should be intact, and should be more it should actually going to facilitate the enzyme to catalyze the reactions. So this is So this is all about the PCR, and I hope you have understood the whole process both by the theoretical explanation as well as by the virtual demos to how the process is being done and how you can be able to perform these two processes in your laboratory. So with this, I would like to conclude my lecture here. Thank you.